One. Hello, my name is Rafal Gutaker, and I am a research leader at Royal Botanic Garden Kew, and I was invited to contribute this talk to Adapt Network. So thank you very much for the invitation. I would like to talk today about plants, which is different from other talks published already, and in particular about potatoes, about their origins and about their adaptation to European continent. So this work has been already published in Nature, Ecology and Evolution, and I need to mention that it was done during my postdoc at Max Planck Institute in Tübingen. It was funded by Max Planck Society and conducted under the supervision of Hernan Bourbano, who's currently at UCL. So I would like to start by introducing a largest resource for any plant biologist, really, which is herbarium. Herbarium is basically a room full of dead plants, which not everyone would be excited about, but a ancient DNA scientist such as myself really makes it the most of this fantastic research. So here in the photograph, I'm showing you in the top one of the oldest and most comprehensive collections of old plants, herbarium of Sir Hans Sloane, that's hosted at Natural History Museum. And in the bottom, you can see an example of one volume of his collection with the plant material still attached to the book. Contemporarily, herbaria look a little bit more like that, where you have just stacks of shelves filled with folders. Each folder would have a number of herbarium sheets, and on each sheet you will have mounted at least one plant, in some case multiple plants. So given the not so old age of those samples, why are herbariums source of my excitement? Well, it's really the answer to that question is in the numbers of samples one can obtain from herbarium. Based on the review by Lang and collaborators, we can see that number of specimens, for example, collected in Europe, reaches 200 million. What that means is that basically, if you name any given species of plant that has been discovered, you're sure to find at least hundreds of historical specimens of that species. In most cases, you will be able to find a single region, geographic region that you're interested in, and have temporal sampling of a plant from this region, which is just an amazing resource. As I mentioned before, maybe herbarium specimens do not really fall on the most impressive side of the spectrum in terms of the age. So if we look at ancient DNA studies, the record holder would be a recently published study by Pechnerova, which sequenced the ancient mammoth from permafrost that was dated back to 1,200,000 years, which beat the record of the previous record holder, which is 700,000-year-old equid, also from permafrost. Outside of the permafrost, ancient DNA is preserved mostly as desiccated or sometimes in sediments, such as highlighted in this study by Sloan et al., where ancient sediments have been sequenced that were dated back to 550,000 to 14,000 years ago. Now back to skeletal remains, desiccated skeletal remains, and entering the realm of sequencing ancient hominins, we have the Proto-Neanderthal sequence that was dated back to 430,000 years old, and hunter-gatherers from Kostenki about 36,000 years ago. So in light of all that, even archaeological studies on plants do not look as amazing. I think one of the oldest specimens ever sequenced was a barley seed that's 6,000 years old. Now, it's really, I think, depends on the tissue that gets preserved. So bones are really remarkable in their ability to preserve DNA. Seeds, which are actually designed to preserve DNA for the future generation, is not nearly as good of a vessel of a DNA, but it gets us at least that far. When we look at the leaf material, so sourced from herbaria, for example, it reaches about 360 years, which is the oldest herbarium full genome sequenced sample from the potato in this study. So should we even call it ancient DNA? I mean, most people would agree that there is very difficult to pinpoint a date, an arbitrary cutoff based on which you would say what is ancient DNA, what's not, say 1,000 years. Instead, a lot of researchers would agree that ancient DNA should be defined by its biomolecular characteristics. So basically, everything that at some point got dead accumulates post-mortem damage to the DNA that really shapes the characteristics of ancient DNA. And the two most relevant reactions that occur in dead tissue is depurination and deamination. Depurination causes fragmentation of a DNA backbone. So in other words, when we sequence DNA of historical or ancient samples, we'll only sequence the small fragments that were preserved throughout the times. So in this case, for example, most of the DNA fragments found in a particular specimen of a herbarium is about 60 base pairs. So it's very, very short. And this is only 150-year-old herbarium specimens. It has been estimated that herbarium specimens, DNA of those, degrade at about 6 to 8 times faster when compared to the ancient bones of moa birds, for example. So really, the fragmentation of the DNA in herbarium is very, very fast. At the same time, in herbarium specimens, or in its DNA, we still observe the so-called damage patterns that are the result of the spontaneously occurring deamination. They're also sometimes called C2T substitution patterns because we see this elevated number of C2T substitutions at first base of each molecule, or maybe not each molecule, but a significant number of molecules. Compared to ancient bones, damage is actually accumulated in herbarium at a very slow pace. So usually we observe about 4% of DNA damage in the beginning, where in bones, this number can reach all the way to 40% or above. Again, slightly different from ancient remains and skeletal remains. In herbarium specimens, we observe a very high proportion of endogenous DNA. So the DNA that actually comes from the species of interest. So this makes work on herbarium much easier compared to ancient bones. But overall, all these three characteristics, so it being fragmented, damaged, and somewhat of metagenomic nature, contaminated with post-mortem colonization of bacteria, fits really the definition of ancient DNA. And so I think historical materials such as herbarium could be still placed under the umbrella of ancient DNA. So now switching gears a little bit and talking about the main point of interest of this paper, which is potato. So we all know potato from the tables and from our plates, but not all of us know much about its biology. So in here, I'm presenting the plant of the potato as a beautiful illustration. Yes, it does have leaves, just like any other plants, not only the tubers. And the interesting fact about potato is that tubers actually are developed not from roots, but from underground shoots. It's called stolons. So in here, I'm presenting you a stolon that slowly swells up into a large tuber. And that's something to consider when I will be talking about my work on potatoes. Potatoes were domesticated about 8,000 years ago, or at least this is the oldest evidence we have from archaeology of the region, and it was domesticated in the Andean region of South America, so in very high altitudes, but low latitudes. Subsequently, in pre-Columbian times, it dispersed to southern Chile, where it was cultivated in much lower altitudes, so basically near the sea, but at much higher latitudes. Finally, about 500 years ago, Spanish colonization efforts brought back spoils of war, potato included, to Europe. In the beginning, it was not used as a food crop, but rather as an ornamental plant. What's really important to realize is how different environment is in all these three regions of the world. Especially important from potato standpoint is the amount of the growing season you get and the variation in day length. So in the Andes, close to the equator, you would have growing season that spans pretty much the whole year. And at the same time, you would have about 12 hours of daylight every day. In these conditions, potato evolved to set tubers only in what we call short days, so these 12 hours of daylight a day. So then if we look at the environment in Europe, where growing season is much shorter, it starts in spring, goes through the summer, all the way to early autumn. This period of time falls under what we call long days, where there is a lot of daylight hours every day. And this is a signal for potato not to start tuberizing. In other words, the potato in Europe will only start tuberizing in short days somewhere in October, and that would be way too late to produce healthy tubers. Instead, the frosts associated with coming winter will kill the plant before we get any delicious potatoes. What's interesting is that environment in southern Chile is quite similar to that in Europe. Again, we have shorter growing season and variable day length across the years that matches this growing season. So it might be that potato already adapted in southern Chile to this, to setting tubers during long days. There is few things we know about the molecular mechanism under that controls tuberization in potato plants. A study that conducted a GWAS on a large population of both long day and short day sensitive plants showed that a single locus could be responsible for as much as 70% of tuberization under long days. They pointed at the gene which is called CDF, and its wild allele is intact and is able to inhibit tuberization during long days. However, a series of inactive variants which have structural variation already allow plant to tuberize under long days. So it has been elucidated further with the whole molecular pathway, where in short days, CDF gene inhibits an inhibitor of tuberization, which then allows potato to tuberize normally. However, in long days, this inhibitory effect of CDF is cancelled, and so potato cannot tuberize. However, the inactive variants, the structural variants of CDF gene, constitutively represses the repressor of tuberization and, in effect, allows potato to tuberize in both short and long days. So this led us to ask two major questions about potato evolution. First, what is the origin of European potatoes? Did it come from the Andes, maybe from Chile, or maybe from somewhere completely else, different place? What is the mode of adaptation of potatoes to European climate? Was it something that evolved from standing variation in the Andes, perhaps standing variation in Chile, or maybe it was a de novo in situ adaptation in Europe? In order to answer these questions, we've reached to herbarium resources, and we sampled 30 samples, historical samples of potatoes, some of which are pictured in this slide. So starting from top left, we'll have a beautiful small leaf that was used for DNA isolation. But then when you look at the bottom left, you will see this very small snippet. That was all we got from one of the oldest herbarium specimens ever collected, which is 360-year-old sample from Sloan Herbarium. To the right, you will see the samples that were collected by Charles Darwin during the voyage of Beagle in South America, and we got our hands on those as well. In total, for European samples, we can observe a spatial distribution in this figure to the right, and the temporal distribution in the bottom. So you will see that we have two samples that are dated to 17th century, then 7 samples that are dated to 19th century, and much higher, much more dense sampling in the recent past. What we did first after isolating DNA is construct the genomic libraries that were without the so-called damage repair. So there was no UDG treatment, just to authenticate if this DNA truly belongs to old specimens. And we found the fragmentation and C2T damage patterns that are consistent with 200 to 300-year-old herbarium specimens. Subsequently, we made genomic libraries, this time with the so-called UDG treatment, so repairing all the C2T substitutions that we used for evolutionary inference. So in here, I'm just showing you a reminder of the 3 major geographic regions we're interested in this story. And here I'm showing you a series of samples that we've sequenced of potato. So majority of which will be representing the diversity in the Andes. and in Chile. However, on the top, you will see 5 samples that represent the earliest introduced potato in Europe. The heat map represents with darker colors closer genetic affinity between 2 different samples. And so you will notice that these 5 earliest introduced European samples are similar to one another, and also more similar to the Andean land races compared to the Chilean land races on the very bottom. We also inquired with what level of ploidy did these early samples have. Potatoes could be diploid, triploid, tetraploid, or even in case of cultivated bitter potatoes, pentaploid or hexaploid. So we thought that if we match the ploidy level of those early introduced European potatoes to any other populations in South America, we could find the source of the introduction. However, it's very difficult to assess ploidy level from something that's dead and doesn't have an intact nuclei anymore. So we can't just use flow cytometry. So instead, we used the frequency of allele calls in our sample to predict what kind of ploidy these historical samples might have had. Just to wrap it all up, if there is a peak of allele frequencies in any given samples around 50%, then that would mean that this sample is most likely diploid. If we observe the 2 peaks at 1/3 and 2/3, then this sample most likely is triploid, and finally 3 peaks would most likely mean a tetraploid sample. Unfortunately, historical samples and their DNA is not usually very good quality data, so they don't give you a very clear answer to this question. But what we can do here is to compare the profiles of likelihood of being diploid, triploid, and tetraploid to other samples that were sequenced in the same study, modern samples for which we had cytometry data. So for example, in this slide, we'll have a 2-axis log likelihood of being triploid versus log likelihood of being tetraploid. And now in faint purple colors, we'll have modern potato tetraploid varieties, which we did flow cytometry on. Now you will see that historical samples cluster together with those. So it most likely means that they were tetraploid as well. So if we look at the ploidy levels again, most likely the earliest introduced potatoes in Europe were of Andean origin from some of the samples that have tetraploidy. We then looked at the chloroplast type, so the whole of the chloroplast genome, which is an equivalent of what people studying animals would use mitochondrial genome for. And we elucidated that the chloroplast type of these earliest introduced potatoes matched some of the tetraploid varieties that originated in the Andes. So that gave us an answer of what the earliest potato in Europe have been like. But the next question we ask is how this potato in Europe changed over the next 400 years. So to that end, we sequenced the whole chloroplast genome for all the historical samples, and we grouped these samples based on their chloroplast type into different clades. So in the bottom, we have a large Andean clade. In the middle, we have the clade of Chilean origin. And on top, we have a small clade that represents a wild relative of potato, Solanum vernei. Now, if we look at frequency of this chloroplast type in time, you will notice that early on, congruent with my previous slide, all the samples had chloroplast type of Andean origin. However, over time, this was replaced with chloroplast type of Chilean origin. And finally, in 20th century, we observed increase of chloroplast type that's matching a wild relative of potato, so the Solanum vernei. We wanted to confirm these results with the diversity we observed in the nuclear genome as well. And so I'm presenting here a PCA plot for all the samples sequenced in this study. So it's a little easier to understand what's happening when looking at dimensions 1 and 3 in the right panel. We have all these green samples clustered together, which represent the diversity that comes from the Andean region of South America. In dark green, we have diploid varieties. In light green, we have tetraploid varieties. Now, going up a little bit, we have 5 of those earliest introduced potatoes in Europe, which are closely related to Andean land races. On the far right side, we have three samples in color teal that represent the Chilean diversity. And you will see that the purple and pink samples that represent diversity of potato in Europe in 18th and 19th century subsequently are intermediate between Andean and Chilean samples. So it could be best maybe pictured with this kind of circle where the first arrow would indicate introduction of Andean potatoes to Europe. Then over time, this would change towards closer to the Chilean genotypes. And then it would go all the way down with a completely different diversity marked with red, which is the 20th century contemporary potato varieties, which apparently are genetically quite distinct. And then finally, the circle will close with introduction of those modern European potatoes back to South America. And so the gray samples are most likely hybrids that are reintroduced from Europe to South America. So in order to scrutinize this data in a little bit more details, we used the population F4 statistic ratio test to understand what's the proportion of Andean ancestry in samples of potato in Europe across time. So early on, we have 100% of Andean ancestry, and then over time, this drops more or less with a linear fashion all the way to 50% of Andean ancestry. You will observe that there is a huge disturbance to this linear pattern that started somewhere in 1840s. And we think that this might be a response of farmers to the potato late blight epidemics that completely decimated potato crops in Ireland, causing 1 million people to die of starvation and further 3 million to emigrate from Ireland. However, this was a problem all across Europe. So really, it was a big change for potato farmers who couldn't cultivate their crops anymore. So we here make a speculation that perhaps they tried to reintroduce their old stocks of Andean potatoes to remedy the situation. Finally, we used the population F statistic framework to reconstruct the admixture graph of our spatiotemporal populations. And so starting from the earliest divergence, you will see that there is a lineage that leads on the top to a ghost population, and on the bottom all the way to the green Andean populations. The second split goes to the Chilean populations in color teal, and then the earliest introduced European potatoes in blue come from direct line of the Andean diversity. But subsequent spatiotemporal populations in Europe already received an admixture from the Chilean lineage. And then in 20th century, there is another admixture event from this ghost population, which we hypothesized to be the potato wild relative that also contributed its chloroplast type to this population and was used in potato breeding in 20th century to bring in resistance to various viruses. So we tested that with a first statistic, and it turned out to be correct. So most likely we observe a gene flow from Solanum vernae. So that answers our question about the origin of potatoes in Europe. So they first came from the Andes, and then there was another wave of potato immigrants coming from Chile, and the resultant potatoes in Europe are actually a hybrid of those two populations. However, we also wanted to understand the origins of this adaptive allele that confers tuberization under long days, so European conditions. In the left, I'm showing you one active allele that blocks potato from tuberizing in long days, the series of 2 structural variants that were already published, and the 4th structural variant with this little green insertion that we discovered in our study. And so in here, I'm showing you the diversity in the middle surrounding the map of Andean and Chilean potatoes in South America. In the middle of all of those circles, you'll see a chloroplast type. So most of the Andean potatoes have the chloroplast type consistent with their origin. In the gray background, you will see pie charts that indicate whether they do have any DNA or DNA reads that support existence of alternative alleles that would confer adaptation to long days. And so in Peru, Colombia, in Bolivia, Argentina, you see no sign of this adaptive allele. You do see a little bit of it in Ecuador, but this is one of those samples that we hypothesize were reintroduced from Europe. Similarly, when we look at historical samples in Chile, the 3 samples from 1850s also had no sign of adaptive allele. This was only discovered in 1950s. However, all those 4 samples we hypothesize were introduced as well because they have very similar genetic signature or footprint to those contemporary potatoes in Europe. So we concluded that it's unlikely, or at least unsampled, we didn't sample the adaptive alleles in South America. So then we looked at the diversity of potatoes already in Europe. So the 1650s oldest introduced potato samples in Europe all do not have the adaptive allele. So again, indicating that this allele did not come from South America. Instead, starting from 1750s, we see increase of DNA supporting existence of at least one of those variants that would already confer the adaptation to long days. So here we concluded that potato adapted within 100 years, most likely in situ in Europe. Finally, there is few historical samples we have from North America, and then these also have all the hallmarks of potatoes contemporary from Europe. All right, so the big question we had afterwards was, for 100 years, potato was not adapted using this known molecular mechanism. So could it be pre-adapted by any other means? So to that end, we did genomic scans by means of the loss of heterozygosity and TGMSD in the bottom to look for outliers. So in other words, we were looking for genomic regions which lost much more diversity than the genomic background, which could indicate operation of selection. And indeed, we found very strong signature, especially in the earliest potatoes marked in blue on chromosome 10, but also a signature that was persistent across next 300 years. We found that the very same region was an outlier for the TGMSD test, indicating that indeed it might have been under selection. We looked at the genes that are annotated in this fragment of chromosome 10, and we found few genes that are responsible for controlling flowering time in potato, but more importantly, 3 genes that are responsible for synthesis of gibberellic acid, which is a phytohormone used by potato to control tuberization. So we concluded that most likely, before potato gained the very effective adaptation through the mutation of CDF gene, it was pre-adapted by regulating its phytohormone economy. So finally, I just wanted to thank you for your attention, acknowledge my previous supervisor, Hernan Burbano, and a colleague from Max Planck Institute, Clemens Weiss, and all the external collaborators that contributed a very important knowledge and samples to this study. Thank you.
I mean, I thought it was very convincing, the evidence you showed for introgression from Chilean, more recent introgression from Chilean land races into the Andean populations, or originally Andean populations in Europe. And I was fully expecting when you said that we're going to see some signatures of introgression of those specific alleles, the CDF alleles, into the European populations. But so it was really surprising that isn't what you think you observed, really, or you just didn't, that was what the data showed. So I wondered what the significance of that shift. Do you think they were bringing in alleles that are useful for other aspects, or for flavor, or something like that?
Yeah, it could be the case. I mean, we were very puzzled ourselves because the emergence of the allele in European samples is contemporary with introduction of Chilean ancestry, but yet we couldn't find any Chilean sample that actually had this allele. So that was very surprising. And one maybe very prosaic reason for that might be that we have very few samples from Chile, and we might have simply missed it. Especially, it was shocking for us to discover that pretty much all contemporary potatoes that are understood as indigenous land races of Chilean origin are actually all replaced by contemporary potato from Europe. So we could only rely on those 3 samples rather than many other samples we had from contemporary, which then of course raises this issue that maybe we just have missed it. But maybe not. And in order to study that, we try to get funding to kind of understand this diversity of potato in Chile in more details, and hopefully then rule out or confirm that it came from Chile. Now, there might be many other adaptations that contributed to the success of Chilean germplasm in Europe. As I said, the environments are very similar, and the day length adaptation is probably the most critical, but there would be many other ways in which Chilean potatoes are more similar to or more fitting to the environment in Europe. It could have something to do with high temperatures during summer, which maybe are not observed in high Andes so much, or any other environmental change. Either way, they seem very, very important for Europeans and somewhat successful. What's also interesting, and I didn't really mention this, is historical musings about the origin of potato in Europe. So one reason why people thought the earliest potato in Europe couldn't be of Chilean origin was based on the fact that contemporary maritime technology did not allow ships to sail fast enough to Europe for potatoes to survive the journey. And so they concluded it must be Andean, because then it was just brought, say, from Colombia to Mexico and from Mexico to Spain in a much faster fashion. So yeah, I just wanted to joke that maybe the Chilean samples were successful in Europe because they were better adapted to maritime journeys. I don't know.
Lost to the mist of time, I suppose, isn't it? Do you think there might be a signal? I mean, if these alleles did come from Chilean origin, do you think there would be a signal in the flanking sequences that might be greater similarity to Chilean?
This is really surprising. This is something we really looked for a very long time, for basically some SNPs that would be in linkage disequilibrium with this insertion and would give us any glimpse into this association. However, what's surprising is that there is almost no linkage disequilibrium. And looking at the number of different structural variants that we observed in this particular gene, it simply might be that there was more than one structural variation event happening. And so we don't observe this kind of flanking sequence pattern that would be interesting for us. So the hypothesis is that there is a site for insertion of a transposon. That's one structural variation. The second structural variant is when this transposon was already excised because it moved further. And the third structural variant that we found do not match this story at all. It must have been an independent insertion of this short fragment. And so we think that maybe this gene has some sort of a hotspot for insertions. So we cannot find that out. What we also try to do is try and time the insertion, kind of figure out whether it happened a long time ago in Chile or, very recently. And this is possible by comparing the two flanking regions of the transposon and seeing how many mutations they accumulate. But again, here we didn't find enough resolution to address that question. So we tried many ways, but yeah, I think the answer would be just sequence more.
Yeah. Well, yeah, I mean, you tried everything and there's no signals, so it seems like the only way. One final question that I was wondering about, that is maybe not that general interest, but I wondered if there was lots of evidence of gene flow between ploidy levels in potato. or if it was all restricted.
Yeah, this is incredibly interesting and well understudied. So people don't really understand the shifts in ploidy levels in potatoes. There used to be used as one of the markers to delineate different species in Andean land races, but people soon realized that they really do not abide by any segregation rule. So hence, we also observe triploid land races. So I think it's very labile for potatoes, their ploidy levels. And what was very surprising in our set is that we found chloroplast type and ploidy levels to match one population in South America, where based on just simply on SNP distance, they actually matched the signature of diploid land races in South America. So we just thought, oh, well, it was just one of these events where diploid became tetraploid because they do it spontaneously and often.
Very interesting. I wondered as well whether, because it looked like the genetic distances were closer to diploids, the European genetic distance closer to the Andean diploids than to the tetraploids. So I wondered if there'd been something funny if they tetraploidize in Europe afterwards. But
yeah, that could be it. Yeah, that's probably. I mean, I wanted to say it could have happened in tubers on the ship, but then I realized you do need sexual reproduction in potatoes to actually have polyploidization events, so it couldn't happen during clonal propagation through tubers.
They're outcrossing, aren't they? They don't self, do they?
No, not too much, no. They are allotetraploids, so I think they're mostly crossing.
Great. Well, thank you very much for a very interesting talk.